This technique is thin layer chromatography, or TLC. For that, we have a glass or plastic plate. We also have a test tube containing some acetone, a long, thin capillary tube, and a paper towel to blot the capillary tube on. You also need a pencil in order to mark up the TLC plates. You start by drawing a line toward the bottom of the TLC plate that runs maybe a centimeter above the end of the TLC plate. Fairly hard to see that, but there it is. So I'm actually going to run two different TLC plates, and therefore I'll mark up both at the same time. What we're going to look at is those two reactions that are running there. I'm trying to run TLC plates to figure out what's going on in those two reactions, and therefore I'll need to have plenty of lanes on this TLC plate. I can look at the two reactions side by side, and I can also look at the other components that go into the two reactions that I'm running. These reactions involve two starting materials. There's a white powder aldehyde that I threw in there, and there's a yellow powder, otherwise known as the ketone, that I'm throwing in there. Those are the two starting materials for this reaction. The aldehyde is starting material 1, the ketone is starting material 2. Both reactions are very similar. They actually contain the same material, but they'll lead to potentially different products, which is why I'm going to run some TLCs. I'm going to want to compare each reaction to each of the two starting materials. In order for me to get solids onto a TLC plate, I need to get them dissolved in some solvent. In order to create a TLC standard, you'd need just a tiny little bit of compound, maybe a speck, a couple of flakes, not much more than that. May not even be particularly visible on camera, but that right there is no more than a milligram or so of the compound, and does not end up amounting to very much at all, but it's more than enough for TLC. So that was the aldehyde, and now I'm going to take another speck or a smidgen of the ketone and make a TLC standard from that. Again, barely visible on camera, if at all. Can hardly even tell that it's there, but that's enough for TLC. Once you have two solids in some container, then all you need to do is add a little bit of solvent. The identity of the solvent doesn't matter. Anything polar that will dissolve things will work. Dichloromethane, making a colorless, essentially colorless solution with the aldehyde will work just fine. Acetone, dichloromethane, either one will work. And it doesn't matter how much solvent you get in there. You just need to get the stuff dissolved. The identity of the solvent doesn't matter because you can just place it on the TLC plate. And once it's on the TLC plate, it'll just simply evaporate. So now we have everything we need, except that we need to mark the TLC plate up into the right number of lanes. So what I'm going to do is I'm going to put both reactions on the plate. I'm going to put this starting material on the plate, and I'm going to put this starting material on the plate. That sounds like 4 different lanes on the TLC plate. But as it turns out, it's really useful to have co-spots. That is, lanes that contain multiple compounds, so that I can do direct comparisons. So what I'm going to do is divide each one of these plates up into 7 lanes. The lab manual does it by drawing little X's. Personally, I prefer to draw hash marks, and I'll put the compounds in between the hash marks. So I have 6 hash marks drawn. That gives me 7 lanes, and I'll go ahead and populate those in a minute. Second TLC plate, same idea, 6 hash marks, 7 lanes. Okay, so in lanes 1 and 7, I want to put the aldehyde compound. How do I actually put the compound on the plate? Simply take the aldehyde and take my long, thin capillary tube and dip it down in there. Very hard to see, but there's actually compound in that capillary tube. And then I just simply touch it everywhere I want this compound to end up. So I want it to end up in lanes 1 and 7, so I touch it to the first lane and the last lane. And on the second TLC plate, I will do the same thing. However, I'm also going to use the aldehyde in lanes 2 and 6 on each TLC plate because I want lanes 2 and 6 to be co-spot lanes, lanes that contain multiple compounds. So I'll go ahead and add the aldehyde to lanes 2 and 6 on each plate. Okay, now that I've concluded adding all of the aldehyde that I want to add, then if I want to, I can rinse out this capillary tube. Simply take a little bit of acetone, dip the tube into the acetone, and then place the capillary on the paper towel, and repeat that a couple of times, and the stuff is clean. Okay, I would like to place the ketone on lane 4. So again, dip it in the ketone, place it on lane 4 in both cases. Just touch it to lane 4, and that should be that. And again, if I want to wash, all I need to do, dip capillary in acetone, touch it to paper towel, and wash. Washing is straightforward. Placing normal compounds onto a TLC plate is fairly straightforward. So this next part is sort of an advanced technique. Essentially it amounts to how do you manage to get a TLC of a reaction that's running under argon gas, an inert atmosphere, while not exposing your reaction to the air. So what you do is you stick a needle through the septum and then just slide the capillary tube down through the needle that you just put in there. And what that allows you to do is pull out a little bit of the reaction mixture from the reaction without exposing it to the air. And then I'm going to add this to lanes 5 and 6. This is actually reaction 2. So touch it to lane 5 and release a little bit of this solvent solution, and then touch it again to lane 6. I'm going to repeat it for the second plate, so I'll do the same thing. Now that I've actually touched the compound to the plate and I've made the tiny little dots, you can tell where I've spotted the reaction mixture because it's highly colored, then it behooves me to actually clean out the spotter between reactions. The whole point of this is to figure out what's in reaction 1 and reaction 2, so I want to make sure that the spotter is clean in between. However, under some conditions, I don't actually care if the spotter is all that clean, so I only clean the spotter sometimes. In order to run TLC plates, what you need is some container to put the solvent in, like a jar, and you also need the solvent itself. I have actually generated a solution of 20% ethyl acetate in hexanes. By personal preference, I like to use a syringe to measure out my volumes. You can use a graduated cylinder or whatever is more convenient. Stand up the TLC plates in the solvent and allow them to run. It's probably easiest to do that with a pair of tweezers. Just make sure that the TLC plates don't fall over in the TLC chamber. And you can run multiple TLC plates at the same time. There's no reason why you can't have many of them going at the same time. Ideally, you cover the TLC plates when they're running, and you simply allow the solvent to move up the plate. And watching a TLC plate run is pretty boring, kind of like watching paint dry. It seems to work better if you just go away for a while. So here comes the solvent up, and we just wait until it gets near the top. Once the solvent gets near the top of the plate, you can pull the plate out. You can see that the solvent line stays there for a little bit, but as soon as you pull the plate out, you just want to mark where that line was. If you blow on the plate, you can actually watch the solvent evaporate off pretty quickly. I'll do the same thing over here. Just simply pull it out, and as soon as I pull it out, mark where that line is. Solvent evaporates off pretty quickly, and now we need to look at the plates. Once you have run the TLC plate, the first thing to do, almost always, is to look at it under a black light. This is kind of interesting in that the two TLC plates should be identical to each other, but I was expecting the two reactions to be more different from each other, and they look pretty similar. But in any event, what you now do is you take a pencil and you circle anywhere you see black dots. So over here and in the co-spot, there's the same spot. Over here, there's two spots. Here's some main spots down over here on the left-hand side, and so on. You simply circle anything that you see. Co-spots should be an exact mixture of the two lanes that are in both of those spots. So over here, now that I'm looking at lane 6, it should be a mixture of lanes 5 and 7, and it appears to be, and so on and so forth. The reason why I ran two TLC plates that were identical to each other comes when we try to stain a TLC plate. So what we're doing right now is visualizing a TLC plate by looking at it under a black light. And the compounds are absorbing UV light, which gives rise to these dark dots. But when I take away the UV light, then I can't see the compounds anymore. And if I would like to have a more permanent sense of where these compounds are, then I need to go ahead and stain them and make the TLC plate permanently colored. So for permanence, I'm going to run two different types of stains. The first is 2,4-DNP, 2,4-dinitrophenylhydrazine. This is actually the test for whether a carbonyl is present. So if I have carbonyls on the TLC plate, I would expect those to show up as red or orange by the time I'm done. So all I do is I take the TLC plate and I dip it in this solution, and then I blot off the excess on the paper. The other stain I'm going to use is ceric ammonium nitrate, CAM, and this is a general TLC stain that just sort of generally oxidizes all sorts of things. Between 2 or 3 different TLC stains are commonly used. CAM is one of them, KMNO4 is another, iodine, and so on. In any event, once you have dipped a TLC plate and rinsed off the excess, blotted off the excess, then you can simply heat it up. So I'm putting it onto a hot plate, and as we heat this up and as we burn these plates, you'll slowly see compounds start to emerge. Everywhere that you see a dot is a compound that is active under a given stain. So what we have over here on the right is the CAM, which is blue, blue dots and yellow dots and so on emerge. And anywhere you can see a compound before pretty much gives rise to dots like you see there. Meanwhile, like I said, DNP is a carbonyl sort of stain, and it is possible to overexpose a stain. As you can see there, it is charred very, very dark. However, it does give me an indication of whether the aldehyde is present or not. Over here on the left is specifically where I can see the aldehyde. So the dark spots on DNP are carbonyls, or at least former carbonyls. So after that particular display of incompetence, I had to go ahead and rerun the DNP TLC plate. And this one is now exposed at a more ordinary level. It's starting to fade a little bit, but these spots right here were initially very pale orange, and these were darker red-orange, indicating that these were carbonyls, and these were possibly carbonyls. Ordinarily, with a DNP stain, red-orange or a red is a carbonyl, and a yellow could be a carbonyl, but it could be other things as well. Likewise, on the right-hand side, what we have now is the cam-stained TLC plate. Focusing more intently on this cam plate now, what I want to point out is that lane 2 is very clearly a mixture of lanes 1 and 3, and lane 6 is very clearly a mixture between lane 5 and 7. Whenever you have a co-spot, unless something is seriously wrong, the co-spot lane should be a simple 50-50 mix of the two lanes that are participating in that co-spot. If it looks different than that, something bizarre is happening. So now, interpretation of the reaction. What does this mean? Well, I had starting material, aldehyde, in lane 1, and it's present in lane 2 as a co-spot, and there's a very, very, very faint trace maybe of it in lane 3, which is the reaction itself, but it's largely gone. For reference, when we looked at the DNP plate, it appears to be gone. So I don't know if it's there or not in reality, but my analysis at the time was that the reaction was complete, and so I stopped the reaction. Likewise, over here on the right-hand side, aldehyde clearly present in lane 7, and in the co-spot, clearly gone unambiguously in lane 5. Therefore, reaction 2 was clearly done because the aldehyde had been used up. As mentioned in the little boxes, the ketone is present in excess throughout the reaction, which means that it's going to be left over. And so the ketone I only placed in lane 4, but it shows up in the reaction mixtures and in the co-spots if you look carefully. In other words, since the aldehyde is gone, the reaction cannot proceed any further. It doesn't matter that there's ketone remaining. Take-home messages from TLC plates. Anywhere you see a spot, that is a compound. The location of the compound, the height of the compound, tells you something about the polarity of the compound. And the assumption is compounds of similar polarity are likely to be the same thing. It doesn't have to be true. In this particular case, since I know what I put into the reaction mixtures, it's a logical explanation. And that's essentially TLC. This is how we use TLCs when we are working up reactions and trying to identify what's going on in reactions.